Hello friends, in this lecture we'll talk about the other aspects of enzymes. Here are some questions that have been asked in the CSIR examination. Indicate the incorrect statement from the following. Allosteric enzymes function through reversible non-covalent binding of allosteric modulators or effectors. Monoclonal antibodies that catalyze hydrolysis of esters or carbonates can be produced. Enzymes are not inhibited irreversibly by heavy metals such as Hg+2 or Ag+ ions. Acid phosphatases hydrolyze biological phosphate esters at approximate pH 5. Then another question was asked in June 2018. Which one of the following statement is not correct? Allosteric enzymes do not obey Michaelis-Menten kinetics. The pre-energy change provides information about the spontaneity but not rate of a reaction. Competitive and non-competitive inhibitions are kinetically indistinguishable. And the fourth statement, kcat upon km of approximately 2 into 10 raised to 8 for an enzyme indicates that the value is close to diffusion controlled rate of encounter. So out of this, we have to find out which statements are not or is not correct. Okay, so let us have a look at some other aspects of the enzymes first, and then we'll come to the discussion on these questions. Principles of catalysis. Now the enzymes, they can exhibit different kinds of catalysis. Catalytic reactions can be exhibited by them. These generally include covalent catalysis, acid-base catalysis, metal ion catalysis. Metal ion catalysis is also referred as Lewis acid-base catalysis. So let us look at this one by one. Covalent catalysis. It involves the formation of a reversible covalent bond between the enzyme and substrate. So these covalent interactions between the enzyme and substrate are temporary, and after the completion of the reaction, the enzyme returns to its native form. Such enzymes bear nucleophilic or electrophilic centers, and the example of such enzyme which carries out the covalent catalysis is chymotrypsin. So chymotrypsin is one of the example of the enzymes exhibiting such kind of covalent catalysis. Acid-base catalysis is another kind of catalytic reaction exhibited by some of the enzymes. It involves proton transfer. So two kinds of acid-base catalysis are observed, specific and general. Specific acid-base catalysis involves the hydronium ions and hydroxide ions act directly as the acid and base groups. Whereas in case of general acid-base catalysis, the donors and acceptors of protons are other than hydronium and hydroxide ions. So in these cases, the ionizable functional groups of amino acid side chains and of cofactors, if any are involved in this, then they may contribute to the catalysis by acting as acids or bases. So the functional groups like imidazole, hydroxyl, carboxyl group, sulfhydryl group, amino and phenolic groups of amino acids, they act as acids or bases. So the example of the enzyme which exhibits acid-base catalysis is the lysozyme, which carries out the hydrolysis of peptidoglycan. Metal ion catalysis, often referred as Lewis acid-base catalysis. Here, many enzymes, they require metal ions for catalytic activity. Now, these enzymes can be metalloenzymes or metal-activated enzymes. Now, what are metalloenzymes? Metalloenzymes are the enzymes which binds the metal ion very tightly or requires the metal ion to maintain its stable native state. Whereas metal activated enzymes, these are the enzymes, they bind metal ions weakly during catalysis. So the level of binding to the enzyme is different in both of these cases. Metal ions can function catalytically in several ways. They can act as electrophilic catalysts, stabilizing the increased electron density or negative charge on a reaction intermediate that can develop during reactions. Metal Metals can also mediate oxidation reduction reactions by reversible changes in the metal ions oxidation states. So that was about the catalysis. Enzyme inhibition is another phenomenon that is observed. So this is exhibited by the inhibitors. Now, enzyme inhibition can be broadly categorized into reversible inhibition or irreversible inhibition. Reversible inhibition, here inhibitors bind to the enzyme in a reversible manner, and so they can dissociate from enzyme inhibitor complex. So the enzyme inhibitor complex is often referred as dead end complex. In case of irreversible inhibition, here the inhibitors bind to the enzyme so tightly that once they are bound to the enzyme, they cannot dissociate from enzyme, thereby leaving the enzyme inactive. So here reversible inhibition can be relieved upon dialysis, whereas irreversible inhibition cannot be relieved upon dialysis of the solution. So when you carry out the dialysis of the mixture of enzyme and inhibitor, then if the inhibitor is reversible one, then the inhibitor molecules can come out of the dialysis membrane. Whereas in case of irreversible inhibitors, the enzyme remains bound to the enzyme. Inhibitors remain bound to the enzyme, and so this binding is so tight that upon dialysis, the inhibitor molecules cannot be removed from the mixture upon dialysis. So this is the basis that the reversible inhibition can be relieved upon dialysis, whereas irreversible inhibition cannot be relieved upon dialysis. So here reversible inhibition and irreversible inhibition is mentioned. So the thing is already discussed. Reversible inhibitors, these can be diagnosed by the effect of inhibitor on Km and Vmax. So what is the effect of inhibitor on Km and Vmax? From that we can identify the reversible inhibitors, and the effects or diagnosis, they are obvious from the double reciprocal plot, which is often referred as LB plot, and these can be the basis for the drugs actions. And reversible inhibitors are used as the research tools in figuring out enzyme chemical catalytic mechanisms. So the reversible inhibition can be of three basic types: competitive inhibition, uncompetitive inhibition, or non-competitive inhibition. A fourth one, mixed inhibition, is often found. So these are defined operationally by their effects on enzyme kinetic parameters such as Km and or Vmax. So what is the effect of the inhibitor on Km and Vmax? Accordingly, they can be identified as either competitive, uncompetitive, non-competitive inhibitors. So here is the figure which indicates the enzyme substrate complex that is formed when the substrate binds to the enzyme, and the competitive inhibitor can be similar to the substrate. It can be a substrate analog. So when that inhibitor is present, it will compete with the substrate to bind or to occupy the binding site on the enzyme. So when the inhibitor is bound, substrate is not able to bind to the enzyme. Here, in case of the uncompetitive inhibition, the inhibitor binds to a site which is different from the substrate binding site. But this site appears when the substrate binds to the enzyme. So upon binding of the substrate to the enzyme, a site is created on the enzyme where the uncompetitive inhibitor binds. So here the uncompetitive inhibitor binds to the ES complex, forming ESI complex. In case of pure non-competitive inhibitor, the inhibitor binds to a site very much different from the substrate binding site. So when the enzyme is free from the substrate or enzyme is bound to the substrate, the inhibitor molecule can bind to the enzyme. But upon binding of the inhibitor to the enzyme, the substrate may bind to the enzyme, but the substrate cannot be converted into product unless and until the inhibitor is removed. So here let us have a look further about the competitive inhibition. In competitive inhibition, you can see the inhibitor can bind, it can bind to the free enzyme and forms EI complex. Now, to the EI complex, the substrate cannot bind, but when inhibitor gets dissociated, then the substrate can bind to the enzyme forming ES complex. So once the ES complex is formed, the substrate can be then converted into product. So here EI is the only dead end complex where the enzyme remains inhibited, and unless and until the inhibitor is not removed, the enzyme cannot convert substrate into product. So here the inhibitor binds in the same site as the substrate. Yes, this site is very much similar to that of the substrate binding site, and so because of that it competes with the substrate to bind to the enzyme. And here the competitive inhibitor increases the apparent Km but does not affect the Vmax. So in this case, the high substrate concentration can overcome the effect of inhibitor. You can see over here that in absence of inhibitor, this is the curve of the LB plot, whereas in presence of competitive inhibitor, the Vmax doesn't change, but Km is changed. So this thing you have to keep in mind. Km changes, but Vmax doesn't change. 1 by Vmax value is same, as both the curves are coinciding with each other. So in this case, high substrate concentration can overcome the effect of inhibitor, because as the substrate molecules are high, they will outcompete the inhibitor molecules to bind to the enzyme. Uncompetitive inhibition, here the inhibitor binds only to the ES complex, and binding site of our inhibitor is created only upon substrate binding. So there is no detectable EI complex, only the free enzyme, then enzyme substrate complex, and the ESI, which is the dead end complex, can be found. So high substrate concentration in this case cannot overcome the effect of inhibitor. If we look at the LB plot, we can get the parallel curves. So this is the curve in absence of inhibitor, and this is the curve in presence of inhibitor. So when the Vmax value and Km value both changes, okay, and the slope of the curve also, it remains same. Slope is same. So in this case, this is the uncompetitive inhibitor. So 1 by Vmax value has changed and minus 1 by Km value also has changed, but the slope remains unchanged. So these are certain characteristics of the uncompetitive inhibition. Pure non-competitive inhibition, here inhibitor can bind either to the free enzyme or to the ES complex, forming two types of dead end complexes, EI and ESI. So whenever the enzyme is in EI form or ESI form, the substrate cannot be converted into product. So only when the enzyme is bound to the substrate in form of ES complex, the substrate can be converted into product. So here you can see in case of pure non-competitive inhibition, when we plot the LB plot, the inhibitor, in presence of the inhibitor, the Km value remains unchanged, whereas Vmax value is changed. Slope is also changed in case of non-competitive inhibitor. So Vm is decreased in case of, in presence of non-competitive inhibitor. There is no effect on Km, and high substrate concentration will not overcome the effect of inhibitor. So here is the summary of the effect of inhibitors on Vmax and Km. So in case of competitive inhibition, Vmax remains unchanged, Km changes. Whereas in case of uncompetitive inhibition, Vmax changes, whereas Km also changes. It rather decreases. In case of pure non-competitive inhibition, Vmax changes, whereas Km remains unchanged. Mixed inhibition, in this case, the inhibition pattern, which is a mixture of the competitive inhibition and uncompetitive inhibition, is seen. But in case of mixed inhibitor, the inhibitor has slightly more affinity towards one of the state, that is either it may have the affinity towards the free enzyme, or it may have affinity more towards ES complex. So In case of mixed inhibition also, it can bind to both the kind of species of enzyme, either free enzyme or ES form, but the mixed inhibitor has more affinity towards one kind of molecule, that is either free enzyme or towards the ES complex. In case of non-competitive inhibition, the inhibitor can bind both free enzyme and ES complex at a equal rate. Okay, so it doesn't exhibit any kind of affinity, non-competitive one. But in case of mixed inhibition, the inhibitor will exhibit slight affinity towards either free enzyme or towards ES complex. So here, in case of mixed inhibition, the inhibitor binds to an allosteric site, which is a site different from the active site where the substrate binds. However, not all the inhibitors that bind at allosteric sites are mixed inhibitors. So here you can see from the graph, if the Vmax is changed and the Km is changed as well as the slope is changed. So when all the three aspects are changed, it indicates that it is a mixed inhibition pattern. So from the graph also, we can just predict what kind of inhibition pattern is it. Regulation of enzyme activity. Regulation by non-covalent modification can be done. This is seen in case of allosteric enzymes. Regulation of the enzyme activity can also be achieved by reversible covalent modification, or feedback inhibition and feedback repression also can result in regulation of enzyme activity. So let us discuss the allosteric enzymes first. Allosteric enzymes are those enzymes whose activity can be changed by molecules which are called as effector molecules, or modulators which are other than the substrate molecules. Modulators exert their effect through reversible non-covalent binding with enzyme on the site which is called as allosteric site, which is different from the active site at which the substrate binds. So allosteric site is also called as regulatory site. Regulatory enzymes for which the substrate and the modulator are identical, these are called as homotropic enzymes. Whereas when the modulator is a molecule other than the substrate, the enzyme is said to be heterotropic one. Modulators are of two types: positive modulators, which enhance the enzyme activity. They are also called as activators. Negative modulators, which inhibit the enzyme activity. They are also called as inhibitors. Binding of modulator causes conformational changes in the allosteric enzymes, which influence their catalytic activity. Enzymes with several modulators generally have different specific binding sites for each. Allosteric enzymes don't obey Michaelis-Menten's kinetics, so this is to be kept in mind. Curve is also different. It is sigmoidal for allosteric enzymes. So you can see over here this graph, it is indicating the pattern of the allosteric enzymes curve. So when substrate and initial velocity is plotted, you get a sigmoidal curve. So this is the sigmoidal curve which is seen in case of allosteric enzymes. The processes which involve the allosteric enzymes, these include negative allosterism or positive allosterism. So here, if the effector binding site alters the shape of the active site of the enzyme, making it an inactive configuration, then such a kind of allosterism is called as negative allosterism. So here you can see the substrate can bind to the free enzyme and to the allosteric site when the inhibitor binds. So inhibitor, when it is in active state and it binds to the allosteric site, it results in the change in the conformation of the enzyme such that the substrate cannot bind to the enzyme. So this is the effect of the allosteric inhibitor on an allosteric enzyme. Positive allosterism, here the effector binding sites that alters the shape of inactive site of the enzyme to an active configuration. So here what happens that when the activator molecule binds to the allosteric site, it arrests the enzyme in the conformation which can be easily accessible by the substrate to bind to the enzyme. So substrate finds it very easy to bind to the enzyme, which in turn is already bound by the positive modulator, or if is or the activator molecule. So activator molecule binds to the allosteric site and makes the substrate easier to bind to the enzyme. So that is how the allosteric activators, they can increase the activity of the enzyme. So here you can see the activator, in presence of activator, such kind of curve is seen, and in presence of inhibitor, more sigmoidal behavior will be seen. Feedback inhibition, an enzyme regulation process in which formation of a product inhibits an earlier reaction in the sequence. It controls the allosteric enzymes. This occurs when an end product of a pathway accumulates as the metabolic demand for it declines. So this end product in turn binds to the regulatory enzyme at the start of the pathway and decreases its activity. The greater the end product levels, the greater the inhibition of the enzyme activity is observed. So you can see over here the end product of the pathway use the feedback inhibition to the of the key enzyme of the pathway. So here B can also give feedback inhibition to the enzyme, which leads to its synthesis from the substrate. In case of feedback repression, the inhibitor molecules, they act as the co-repressors and they repress the synthesis of the enzyme that is required at an early stage of the pathway, and that is called as feedback repression. So here the inhibitors, they act at the genetic level. Then protein modification or the covalent modification can also be seen as a regulatory mechanism. So here this is a process in which a chemical group is covalently added or removed to the protein or to the enzyme. So phosphorylation is an example in which phosphorylation of the enzyme can turn it into an active form, whereas dephosphorylation can result it in its conversion to inactive form. The example is that of pyruvate dehydrogenase enzyme, which in phosphorylated form is active, and when it is dephosphorylated, it becomes inactive. So kinase will make it active one, and phosphatase will make it inactive. Proenzymes or zymogen, the inactive form of enzyme which can be activated by removing a small part on their polypeptide chain. Mostly these are digestive enzymes and blood clotting enzymes. So why is it that digestive enzymes are inactive state before it becomes active? This is necessary to prevent digestion of pancreatic and gastric tissues. So here pepsinogen is the zymogen, which is then converted into pepsin. Trypsinogen is converted into trypsin. Prothrombin is converted into thrombin. So these are some examples of zymogens. So you can see the several enzymes, they are present in inactive form and they are made active by the action of other enzymes. So entropeptidase converts trypsinogen into trypsin. Trypsin in turn converts chymotrypsinogen into trypsin. It converts the pro-elastase into elastase, pro-carboxypeptidase into carboxypeptidase, and it also converts pro-lipase into lipase. Isoenzymes is also another category. So the multiple forms of the enzyme catalyzing the same reactions are called as isoenzymes, and they differ in their physical and chemical properties, which include the structure, electrophoretic and immunological properties. Km and Vmax values may also be different. pH optimum, relative susceptibility to inhibitors, and degree of denaturation also differs among these isoenzymes. So let us have a look at the example of an isoenzyme, lactate dehydrogenase. Several forms of lactate dehydrogenases are found in the tissues, different tissues. So LDH, whose systematic name is L-lactate NAD plus oxidoreductase. It catalyzes the interconversion of lactate and pyruvate. So lactic acid is converted into pyruvic acid by lactate dehydrogenase enzyme, and pyruvic acid is converted into lactic acid by the same enzyme. So LDH has 5 distinct isoenzymes: LDH1, 2, 3, 4, and 5, in which LDH1 has more positive charge, and so it runs fast in the agarose gel or in the SDS page, whereas LDH5 is the slowest one. Structure of LDH enzymes. So you can see over here in this table, LDH enzymes, they have the different monomers or of same monomers. The hard H4 is the configuration of LDH1. It consists of only H type of subunits, whereas LDH2 has M and H type. So H3M is the configuration of LDH2, and so on. So circular one are the M or muscle type of monomers, whereas H indicates the hard monomers. So you can see over here, H4 is the configuration of LDH1, and LDH5 has all the monomers of M type. So M4 is the configuration of LDH5. So LDH1 is found exclusively in heart and RBC, whereas LDH2 is found in heart and RBC. LDH3 is found in brain and kidney. LDH4 is found in liver and skeletal muscles, and LDH5 is also found in skeletal muscles and liver. So you can see the percentage of these LDH enzymes in the normal serum, and this is constant for a healthy individual. Now these may change. This is the this is the profile, electrophoretic profile of the LDH enzymes, and you can see this is the of a normal person, whereas in case of myocardial infarction, the LDH1 levels increase and LDH2 levels also increase. Whereas in case of the liver damage, the LDH5 enzyme concentration is seen to be at a higher concentration in the serum. So significance of differential catalytic activity. Here LDH1 is predominantly found in heart muscle and is inhibited by pyruvate, the substrate, and hence the pyruvate is not converted to lactate in cardiac muscle but is converted to acetyl CoA, which enters into the citric acid cycle. LDH5 is mostly present in skeletal muscles and the inhibition of this enzyme by pyruvate is minimal. Hence pyruvate is converted to lactate. Further, LDH5 has low Km while LDH1 has high Km for pyruvate. The differential catalytic activities of LDH1 and LDH5 in heart and skeletal muscles respectively are well suited for the aerobic and anaerobic conditions prevailing in these tissues. This is we have already discussed. So let us have a look at the questions. Indicate the incorrect statement from the following. Allostatic enzymes, they function through reversible non-covalent binding of the allostatic modulators or effectors. Monoclonal antibodies that catalyze hydrolysis of esters or carbonates can be produced. Yes. Enzymes are not inhibited irreversibly by heavy metals such as Hg+2 and Ag+. Acid phosphatases, they hydrolyze biological phosphate esters at pH 5. So here the incorrect statement is this one. Hg+2 binds to the proteins very tightly, and so it can act as a irreversible inhibitor. So the correct answer is 3. The next question is to identify the statement which is not correct. So allostatic enzymes, they do not obey Michaelis-Menten's kinetics. It is true. The free energy change provides information about spontaneity but not rate of reaction. It is correct. Competitive and non-competitive inhibitions are kinetically indistinguishable. No, from the graph you can detect whether it is competitive or non-competitive. And this is the kcat upon km value, which is at 10⁸. So it is indicating that the enzyme indicates that the value is close to diffusion control rate of encounter. So this is correct statement. So incorrect one is the third one. So this was about the enzymes. So thank you and happy learning.